All right, to our audience, welcome to Medical Grand Rounds at the University of Colorado. Thanks for joining us again this week. Appreciate everyone being here. We'll talk for just a second, do a few introductions, and I'll get to our speakers. I always start with big announcements, and I think today we have probably one of the biggest announcements a department can have, which is that we have a new chair of medicine. So I wanted to give just a brief chance for Dr. Chopra to introduce himself and say hello to everybody, and I know he'll have a chance to do that more personally in the upcoming weeks, but Dr. Vineet Chopra is coming. He was previously the chief of the Division of Hospital Medicine at the University of Michigan Health System, and not only was he a chief there, he was the founding chief for that division, one of the newest in the Department of Medicine in the last 50 years. He's an accomplished physician scientist. He's a health science researcher, does a lot of his work focused on patient safety and hospital-acquired complications. Dr. Chopra, welcome to Colorado.
Dr. Karnas, thank you for that very kind introduction. It's a pleasure to be here. I am excited. I'm learning more about our department and the many strengths that we have. And I'm particularly proud today to be here for my first grand rounds with Dr. Smith and Dr. Fry. So thank you for the warm welcome to the CU family and community. Looking forward to meeting you. And thank you, Dr. Smith and Dr. Fry, for making my first grand rounds here a memorable one. Looking forward to it.
Thanks, Dr. Chopra. I want to remind everyone that Grand Rounds, as similar to last year, does qualify for mock and CME credit. And so please use the messages that will come through the chat function to sign up for that. Everyone who attends all of the Grand Round sessions this year could receive up to 40 mock credits. So it goes a long way toward what we need to accomplish. I want to also remind people to put questions into the Q&A section at the bottom of the Zoom, not the chat section. That way the chiefs and I can gather those, and we'll make sure to ask questions of Dr. Fry and Dr. Smith at the end of the talk, and we'll try to get through as many of those questions as possible. And so as a way of introducing our speakers, since there are two today, I will be slightly brief and not cut into their time. I first want to introduce Dr. Clay Smith. Dr. Smith did his medical training at Southwestern Medical School down in Dallas. He was an intern at Parkland and then a resident at Cornell Hospital in New York. He then stayed in Manhattan for his fellowship training at Memorial Sloan Kettering, and then did a bone marrow transplant fellowship in addition at Stanford before coming to the University of Colorado. He is currently a professor in the Department of Medicine. He's the director of the Blood Disorders and Cellular Therapies Center in the UC Health System. He's the associate division head for clinical affairs in the Division of Hematology, and he's the medical director at the University of Colorado and UC Health Innovation Center. And someone else I would like to introduce, which is who is the person who's going to give the first half of the talk, actually, is Dr. Terry Fry. Dr. Fry did his medical school at Georgetown University in Washington, D.C. He did a pediatrics residency and pediatrics chief residency also at Georgetown before going to Johns Hopkins for his pediatric hematology fellowship and oncology fellowship. He was then a postdoctoral fellow at the NCI in Bethesda. He spent a large chunk of his career at the NCI and joined us here at the University of Colorado in 2018, where he is a professor of pediatrics, hematology, and immunology. He's the co-director of the Human Immunology and Immunotherapy Institute. He's the director of cancer immunotherapy at the University of Colorado School of Medicine, and he currently holds the Robert and Kathleen Clark Endowed Chair in Pediatric Cancer Therapeutics. These two high-ranking and highly published professors are going to present today about CAR T cells, cancer immunogenetics, and the future within personalized medicine. And so I will turn it over to Dr. Fry.
Thank you, Dr. Connors and Dr. Chopra. It's really a pleasure today to give this grand rounds, with my partner in the cell therapy program, Clay Smith. And what we're going to do today is to run through. I'll run through in the beginning sort of a background on CAR T cells, going through some of the early developments, and then Clay will talk you through the clinical experience with CAR T cells thus far, give you some exciting updates on the cell therapy program at CU Anschutz. And then at the end, I'll circle back and just spend a few minutes talking about what the future of this innovative therapy might look like. So just as a way of going, way back, the concept of using the immune system to treat cancer is actually quite old. The first description was really this that many of you might have heard about, which is Cooley's toxin. It was an approach that was developed by a physician by the name of William Cooley, who was working at a hospital in New York that eventually became Memorial Sloan Kettering. He was a bone surgeon. One of his claims to fame was he took care of a friend of J.D. Rockefeller's, a young woman with an aggressive sarcoma, ultimately died. But one of the observations that he made was that in some of the patients that he treated with incomplete resections of their sarcoma, he noted that if they developed erysipelas, a postoperative infection, that he occasionally saw these spontaneous regressions. And so he embarked on this trial, a rather crude trial by today's standards, where he essentially injected these sarcomas postoperatively with essentially pus from patients. And he actually had responses, but as you would imagine, they were quite unpredictable. And what happened was that at the same time, there was another physician by the name of James Ewing, who was a pathologist who gave name to a sarcoma called Ewing sarcoma, was just beginning to develop radiation therapy, and the responses to radiation were more predictable. Sort of a bit of a controversy ensued, and ultimately, Cooley's approach sort of died away until ultimately, we began to understand the immune system sufficiently to employ it more effectively. This is a quote which I think is a really interesting quote from one of William Cooley's early papers, which is, nature often gives us hints to a profoundest secrets. And this really, harkens back to his initial observation about the spontaneous regressions and his ability to capitalize on it. This is a picture of one of the patients that he treated. So the other thing I want to do is to just take a step back and just give everybody a bit of a primer on T-cell immunology, at least the aspects of this that are going to be important as we talk about CAR T-cells. So, as you all know, T-cells are activated through an antigen receptor called the T-cell receptor. This receptor endows T-cells with the ability to very specifically recognize peptides derived from proteins in the context of self-HLA. And that's the primary signal that drives T-cell activation. But what we now know is that signal in and of itself is not sufficient, and that the ultimate signal in a T-cell really is a combination of both the signal one that's derived from the HLA and peptide, as well as a number of co-stimulatory signals, both positive and negative signals, that control the activation state of the T cell. And I've listed sort of a set of those molecules here. So one of the early, concepts that emerged in cancer immunotherapy was sort of the notion that if you could shut down the brakes on the immune system, that is, these negative regulators of the immune response, that perhaps you could unleash an immune response against a tumor. And so, really the first proof of principle of this emerged in work from Jim Allison, who ultimately was granted the Nobel Prize for this work. And this was, one of his earlier publications from 1996, in which his team demonstrated regression of large, bulky tumors in mice through blockade of a receptor called CTLA4, which is one of these negative regulators of the immune response. And so, based on this work, this therapy was eventually, turned into, therapeutic in patients. And what's shown on the right was one of the earliest reports of the effectiveness of what is now called immune checkpoint inhibition in the context of melanoma with anti-CTLA4. This is an example of a patient who had regression of both pulmonary as well as central nervous system metastasis through blockade of CTLA4. I think, most of you probably are familiar now with the fact that there's been an explosion of these immune checkpoint inhibitors, most of them now targeting a molecule called PD-1. And there's been a number now of FDA approvals with this approach in adult epithelial cancers primarily. The other thing that's emerged from this work is sort of the observation that obviously not all patients respond. And this is true even if you look within a specific cancer subset, in this case, non-small cell lung cancer. And Jed Walchuk and his team, 2018, began to work along with other investigators at understanding why there was this differential response across different patients. And one of the hallmarks, or one of the findings that emerged, was that the mutational burden of the tumors was at least somewhat predictive of the ability for these checkpoint inhibitors to generate responses in patients. And this is just looking at response to checkpoint inhibitors in cohort of patients stratified by where they're at in terms of mutational burden. As you see, the patients that are in the 75th to 99th percentile mutational load had a higher likelihood of benefiting from immune checkpoint inhibitors. There's certainly a lot of other parameters that define response to immune checkpoint inhibitors, but this is one that was identified early on. The problem is that that many tumors that we deal with are very low in their mutational burden on average. And so on the right-hand side in blue those are the cancers that have typically been most responsive to immune checkpoint inhibitors. The left, the tumors that we deal with in pediatrics, quite frankly, as well as many of the hematologic malignancies, have relatively low mutational burden. And for this reason, as well as others, the experience with immune checkpoint inhibitors, certainly in pediatric oncology, has been quite disappointing. And so, there's been a lot of enthusiasm about trying to come up with alternative approaches to activate the immune system to treat malignancies that don't respond to these immune checkpoint inhibitors. And so one way to do that is through the process of redirecting the specificity of T cells. So on the left is what I've already shown you, which is sort of the standard T cell receptor, which again generates specificity for peptide. And what we, essentially, with this redirection of specificity, the assumption is that in the cases of these tumors that evade the immune system, that the T cells are unable to, quote, see the tumor. And so one way to make those tumors, those T cells see the tumor, is to genetically modify them so that we can control what they see. And so the concept of a chimeric antigen receptor is really an artificial gene that we can introduce into T cell that drives expression of this protein, this receptor on the surface, that is really a hybrid molecule that contains antibody binding domains, the scFv on the extracellular portion, and it links that to standard T cell receptor signaling machinery, signal 1 and signal 2 in the same receptor. Again, a bit of a Frankenstein molecule, that is able to generate specificity that is free of self-MHC and really drives all of the signals for full activation in a single construct. So just by way of background, really the first reports of such synthetic receptors were in the late 1980s. These were the two original papers, the first one in 1987 and then a follow-up by Zeligheshar in 1989. Amazing papers, hand drawings of these synthetic receptors, but this was really the original concept. Fast forward from this original description to 2010, when Jim Kokendorfer really provided the first case report of the dramatic responses that can be seen in patients receiving a chimeric antigen receptor targeting CD19, a molecule that's expressed on B-cell malignancies. And so this is a patient with lymphoma, again, who had refractory lymphoma, who had a complete remission to a single infusion of these genetically modified T cells. And so with that background, we're going to turn our attention now to where we're at in terms of the clinical experience with CAR T cells thus far. Really the first clinical experience was actually in pediatric leukemia. With treatment using CD19 CAR T cells at a number of institutions around the United States. And remarkably, this approach led to an 80 to 90% complete remission induction rate in chemotherapy refractory children with ALL. And that led to, based on the data I'm showing here from the Eliana trial, the first FDA approval for CD19 CAR T cells, which again was a pediatric approval. And then this was followed up then with approvals in adult lymphoma. And so with that, I'm going to turn it over to Clay, who's going to talk about the lymphoma experience.
Thanks. So if those of you noticed, there were 30 years between those first examples of experiments in this field and the proof of principle. And that 30 years was a lot of trial and error, things not working, but mostly it was an incredible testament to the persistence of people like Terry and Steve Rosenberg and others that believed in this despite a lot of obstacles. And what we hope to show you now in the clinical portion of this talk is what a revolution that we now have that's exploding, before our eyes once we finally got through that 30 years of difficult challenges, and now really a lot of refinement and evolution of this therapy. So I just wanted to give a quick typical case of one of our patients. This was a 62-year-old male diagnosed with diffuse large B-cell lymphoma, got treated with R-EPOCH, which is a typical treatment for this lymphoma, get 6 cycles of that, went into remission, but 6 months later, his disease came back, and you can see in the PET scan on the right, before CAR T-cell treatment, all of the disease there in the GI tract and the abdomen. He was treated with one of the CAR T-cell products, and I'll tell you more about the different products in just a minute. Was complicated by a toxicity called cytokine release syndrome a few days after receiving the CAR T-cells, and then was further complicated by neurologic toxicity, which is called ICANS, a few days later, and we'll go into both of those in a minute as well. Got treated appropriately, both of these resolved, and as you can see, 29 days after treatment, this was his disease was in remission. What I'm sorry I left out is that he was also treated with additional chemotherapy after the EPOCH and had failed that. And in those patients who failed 2 rounds of chemo, not a bone marrow transplant candidate, the overall response rate typically with other chemotherapy is very low. So this is really, really remarkable. This is somebody that a few years ago we probably would have been talking palliative care rather than cure. This person continued to be in response at a year follow-up. These treatments are only several years old now from demonstration of initial efficacy. And so we don't know yet whether these folks are cured yet or not. We're going to have to follow them for a number of years more to see it, but it's pretty exciting, as you can see. Next slide, please. So just to walk you through the specific steps of how we do this, a patient comes in and they undergo a process called leukapheresis, which is where we collect their T cells from their peripheral blood. That gets sent off to a cell therapy laboratory, which has to operate under very strict, what are called GMP conditions, where things are very highly regulated and controlled. I think this is one of the remarkable parts of this story is that we now use frequently what are called lentiviral vectors, which are really highly genetically modified versions of HIV, where all of the disease-causing elements have been genetically engineered out. But the part of HIV that allows it to efficiently infect T cells are retained. There are other types of vectors and a lot of work going on in this field, but that's, I think, one of the more interesting stories of taking something that was the COVID of its time back in the 80s and turning it into a force for good. So once the cells have been genetically modified, they are then expanded and grown in that culture and then brought back to the patient. Terry, can you just click through the animations, please? So this just shows some of a typical kind of machinery for some of the portions of this. And just click one more time, Terry. So this comes back. You can see in this, it looks like a transfusion of platelets almost, but that's a bag full of ex vivo expanded genetically modified T cells. These then get infused intravenously back into a patient. And then in the upper right, this is a really cool picture of what those immune effector cells, those genetically modified T cells, physically attaching to a cancer cell and ultimately destroying it, leading to those clinical and PET changes that we just showed you. Next slide, please. Just to point out, we're talking, Terry and I are mostly talking about CAR T cells today, which he nicely described, but there are many, many variations on this theme where people are taking different type of immune effector cells, these including NK cells, these include T cells that come directly from a tumor themselves. Those are called tumor infiltrating lymphocytes or TIL cells. They can be genetically modified by the hybrid molecules, the CAR, the chimeric antigen receptor that Terry described for you, or modified by a variety of other types of molecules that also provide specificity. And there are many people around the world working, in fact, on using CRISPR-Cas and other really cutting-edge mechanisms to try to eliminate the business parts of T cells and other immune effectors that cause things like graft versus host or rejection of the T cell, so that ultimately we can move from this autologous one patient, one T cell, one product model that I just showed you to a more off-the-shelf model where you order these up almost like you would any other medication. Next slide, Terry. In adults, primarily the CAR T cells have been directed at hematologic malignancies for reasons that I'll discuss in just a minute. These you can see over in the upper right, the hematologic malignancies include adult ALL, like the pediatric counterpart. Most of the work has actually been done in non-Hodgkin's lymphoma because that's the most common of the hematologic malignancies that we deal with, and it has this unique target of CD19, CD20, 22, and others that allow people like Terry to engineer these CARs so they're specific for the cancer cell and don't interact too much with normal tissues. BCMA is another target that's found on multiple myeloma cells that there's been some exciting work. There's been a lot of work in my particular field in AML with a variety of targets. And I would say for the most part, those have been disappointing so far, and still much work needs to be done and much improvement there. Next slide. This just shows you again an interesting slide, which is the spectrum of targets that people are developing CARs to. But it also shows where these CARs are being developed. And you'll notice in blue is China, and in the more reddish part is the United States. And China actually at the moment has more CAR trials than the US. And so is really doing a lot of the leading work in the world right now in this area. Next slide. So give you a couple of a little bit more granularity about 3 or 4 of the diseases that we take care of in the adult program and that we've deployed CARs against. The first is B-cell non-Hodgkin's lymphoma. This is like the case report I just showed you, presents average age as patient in their 60s, but can happen in all ages. People come in with lymphadenopathy, like you see, a biopsy is done and you find lymphoma. Lymphoma is complicated by the fact that there's at least 60 different subtypes, and every year it seems like they find more subtypes. The one that's the most common is the case report I showed, diffuse large B-cell lymphoma. And about 60% of these people can actually be cured with chemotherapy. The other 40%, the current standard of care is a autologous stem cell transplant, but many people fail that, or they cannot get into remission, which you need to be in before a stem cell transplant, or they're simply not fit enough to do a transplant. And that's the primary setting that I'm going to show you today for CAR T cells in adults. There's been very interesting work in multiple myeloma. That's about 35,000 cases a year. This remains an incurable cancer, and every treatment we have is really designed to prolong life rather than cure the disease. The good news is there are many treatments for this disease, immunomodulators, proteasome inhibitors, antibodies, particularly against a surface target on myeloma called CD38, the same type of autologous stem cell transplant that we use for lymphoma. But as I said, not curable. The 5-year overall survival is about 50%. And frequently what we do is string one of these treatments after another, like beads on a chain. And again, CAR T cells come into play after several of these more standard treatments have failed. Similarly, AML cure rate still remains about 25%, unfortunately, despite chemotherapy, targeted therapies. And again, for AML and ALL, we typically bring out CARs when people have failed multiple lines of therapy. Next slide. So this is an example of one of the seminal trials of a CAR T cell and I would caution anyone against trying to pronounce the generic names of these CAR T cells. I hope Terry can do it. I still struggle with them. So this is one of the CAR T cells that was developed several years ago. And you can see the overall response rate, the overall survival is around 40% in all patients. And what this also shows you, though, is the people that achieve those complete responses that I showed you at day 29 PET scan, they may have very excellent long-term survival. Now, again, we're limited because these are brand new therapies. The follow-up for some of the earliest patients is really only about 36 months. So we don't know whether this plateau is going to stay for 5 years, 10 years, forever, or are we going to start seeing late relapses, and only time will tell. Next slide. This is a similar type of data from a couple of different studies published in the New England Journal of Medicine relatively recently in multiple myeloma. And again, you can see that there is a response rate. Unlike lymphoma, so far, I don't think we've seen a plateau. And in general, I think we view CAR T cells along the same lines as we do with other therapies in myeloma. Not at all clear whether they're going to be curative, but they do seem to prolong life by another year. And in our hands, we've seen some really exciting responses with people that otherwise would have been going to palliative care that got a year or more out of life from this type of therapy. Next slide, please. These are a list of the CAR T cells that are approved right now. There's the most recent one was Tecartus, which was approved for adult ALL a few weeks ago. There's several hundred companies out there right now working on CAR T cells with variations of these different types of modifications that we've been describing today. But these are the initial FDA-approved commercial products. Next slide, please. So every treatment has its side effects. And as I mentioned in the case report, the two big side effects of CAR T cells and other immune effector cells are cytokine release syndrome. And so what this is thought to be due to engagement of the CAR T cell and then activation of a lot of pro-inflammatory cytokines, which then cause everything from mild fever all the way up to very severe hypertension, hypoxia, capillary leak, and even death in some patients. ICANS is the sort of technical term for the neurotoxicity. Nobody actually knows what causes this. It's an interesting clinical phenomena. We've seen people that have lost just the ability to find words and everything else is intact, all the way up to coma and seizures. And this can also ultimately cause death in a small number of people. Next animation, please. The treatment for CRS is an anti-IL-6 antibody approved by the FDA for this called tocilizumab. And if that doesn't work, we add steroids. For ICANS, it's not clear that the anti-IL-6 agents actually work, and there's some thought that may make things worse. And so we typically start out with steroids. And then, as you can imagine, there is a whole host of other cytokines and inflammatory proteins that may play a role. And there's a number of different agents like anakinra directed at IL-1 and others that are either being explored or we use when other treatments fail. Next slide, please. So I'm going to talk for a few minutes about cell therapy at CU Anschutz, what we've been doing on this campus, and then turn it back over to Terry. Next slide, please. So I think the first and most important thing to know about the CU UC Health effort in this area is it is an absolute team effort. The hospital UC healthcare system is where we perform apheresis and collection of those stem cells. We do all our inpatient care, ICU care, outpatient care, many other activities for these patients within UC Health. And on the CU side, of course, that's where the home for MDs and APPs, lots of research, clinical trials, and others. Next slide. And we have on campus a couple of really important resources. The first is the Gates biomanufacturing facility. This has been built out specifically to manufacture cell therapies on campus. And we've now treated, I believe, 7 patients with CAR T cells manufactured with a vector from Terry. This has been a very, very promising and exciting, both because it opens the door to a lot of exciting science, which Terry is going to talk about in just a minute. That's going to be CU specific. But it also, I think, maybe even more satisfying, allows us to treat patients who otherwise did not have the insurance coverage necessary to get a commercial product or did not fit into one of our clinical trials. And so this has been very, very exciting for us to be able to offer this cutting edge therapy developed at CU for the underserved and people that otherwise could not receive CAR T cells. Clinimmune is another very important operation on campus, which coordinates a lot of the receipt and distribution and storage and quality assurance of these CAR products. Again, highlighting what a team effort this is. Next slide. There's if anybody wants, there's a very nice story to take it from all of the Kaplan-Meiers and statistics I've shown you down to a very human level. This is the story of a patient that was treated by Dr. Kamdar in our group and her journey through the CAR T cell process. Next. So these are currently all of the adult commercial products at the top, and then clinical studies that are being offered through the Hematology Clinical Trials Unit down on the bottom. And you can see, again, most of these are for lymphoma, myeloma, and AML, as well as a small number of ALL patients. Next, please. This is our experience so far. We've now treated about 100 patients. A couple of the patients got 2 infusions. About 100 patients. Most of those, as you can see, are with non-Hodgkin's lymphoma. And if you look on the right, this gives us the overall survival rate, which is at least as good as what has been published in those New England Journal of Medicine and other papers. This has been about half from commercial products, but a half from clinical research. And as you can see, patients that were treated on that investigator-initiated trial through the Gates Biomanufacturing Center. Next slide. So I think I've mentioned before, but I can't say this enough. This is a huge team effort. It requires medical expertise. We have an enormous amount of regulatory guidelines that are actually really excellent that we follow. If you look at that picture on the lower right, those are Lee Gates and our quality team presenting some of the documentation that we submit for FACT and other regulatory requirements in our field. We have a lot of written guidelines for each of these CAR products. You need to go through what's called a REMS program to make sure you know how to treat CRS and neurotoxicity. We have a very robust adverse events and QA process. And a lot of these are led by John Gutman, one of our physicians up in the other left, along with Heather Knowles, who is an APP, but very fortunately also has a PhD in immunology. And she leads a lot of our in-house efforts to provide high quality care. This is also our pheresis team, which shows you how many people it takes to collect these T cells, and then our inpatient team. And of course, we couldn't do any of this without a lot of expertise in nurses and pharmacists and others that make all this tick. Next slide. So these are a very exciting advance in care, but as you can see, there's still a lot of challenges that we need to address. We still don't know where CAR T cells belong in the sequence of treatments. There's some exciting work that I'll show you in a second, showing that it might actually start to replace some of the current standard of cares and move up earlier into the disease treatment course. We have big questions about who's going to respond or not respond to these. Terry's going to talk a little bit about this, but in general, going into a CAR treatment, we do not know the answer to that. And so we treat everybody, even though 60% of people or more may not benefit. It's a month in the hospital for many people. The costs can be up to $1 million per treatment. The CAR T cells themselves can cost $300,000 to $400,000. And the worst of all is, of course, we delay getting people to treatments that might work. Solid tumors, you haven't heard much about that from me, and that's because these are still in early stages. There's a lot more challenges for them because their antigen targets are either difficult or they are shared with normal tissue, and so it is more challenging to generate CAR T cells and other immune effectors that are both effective and specific. I've mentioned already that we would like to be moving from this one patient, one CAR T cell model to being able to order these like a drug. We would, of course, love immune effectors and CAR T cells without CRS and ICANS. And as I mentioned, the cost is still quite prohibitive, and that needs to come down as well to make these widely effective and available. And then, of course, the big question is, how do we combine these with other cancer therapies? As you can probably guess from Terry's earlier comments, people are now testing these with checkpoint inhibitors and other immune modulators to see if they can improve their duration of activity and degree of effect. Next slide. So I'll just show you a couple of these examples of how these challenges are being addressed. Dr. Manali Kamdar in our group, who heads our lymphoma program, has been one of the key leaders in a trial called the Transform trial, which is looking at CAR T cells versus autologous stem cell transplant for those lymphoma patients that relapse. There's another similar trial going on as well. The preliminary results of both of these are going to be announced at the American Society of Hematology in December. And Manali played such an important role in this, and I think her program was the largest contributor to this trial in the world. And so she will actually be presenting this. It's going to be one of the real practice-changing talks and clinical trials in our field in many years. And Manali will actually be the one presenting this to the world at large in a couple of months. Next slide. I mentioned we'd love to know upfront who's going to respond and who's not going to respond. Steve Baer in our group is working with a number of people over at Children's to develop a precision medicine approach where we're looking at both clinical features as well as genetic features in tumors, in CAR T cells, in patients and their immune cells, and then using advanced informatics techniques like machine learning to try to define upfront before someone ever gets treated, are they likely to respond or develop toxicities? And if they're not likely to respond, for example, can we identify new targets to help direct them to better forms of therapy? Next slide. In the solid tumor world, we're very lucky to have Dr. Bree Wilkie, who's leading a trial in sarcoma with also immune effector cells. This is a design of the trial. This is, again, a very exciting area, and I think solid tumor is poised for the same type of explosion and advances that we've been fortunate to see in the hematologic malignancies in the last few years. And then next slide. We also have some very exciting work in the Division of Medical Oncology by Eduardo De Vida and his group, where he's developing a new type of immune effector, which he has named a mighty TIL. So that, as you can imagine, is a tumor infiltrating lymphocyte where he has created a new chimeric type molecule, sort of analogous to the CAR T cell, but hopefully one that will allow the recognition of new types of antigens and boost signals within the TIL cells so the responses are more pronounced. And there's a number of theoretical potential advantages to this. Eduardo has a number of other really interesting, exciting work in his lab going on, and I highly recommend anybody interested in that, you can look at his lab's website at the URL at the bottom of this slide and get a flavor of the really very cool things that he's working on. Next slide. So I'm going to turn it back to Terry, talking about the future of CAR T cells.
Yeah, thanks, Clay. And I'm going to spend about 10 minutes or so, leaving time for questions at the end. Circling back and to the challenges that Clay highlighted, and I'm going to focus primarily on the challenges that relate to the CD19 CAR T cells. I won't talk much about the extensive challenges in other types of cancers that Clay highlighted. But if you look at Clay mentioned that the long-term, that is maybe at a year or so, durable remission rate in patients with lymphoma is about 40%, right? 40 to 50%, probably 40 is more accurate with the real world experience. That means that 60% of patients ultimately, won't enjoy a long-term remission. And if you look at sort of, what happens in a patient with lymphoma who receives a CAR T cell, really there are, three scenarios. One is there's a subset of patients where there's really no response, there's progression. That's a relatively small subset. There's a subset of patients where there's a partial response. And we know that all patients who don't achieve a complete response by 90 days, ultimately those patients will progress. And then there are the patients who respond. And as Clay said, the durability is quite good in lymphoma, but there's certainly a subset that will ultimately relapse. I will tell you that in leukemia, the experience is quite different. I mentioned earlier with the Eliana trial, the licensing trial for Kymriah, the first FDA approval, that the remission induction rate was around 80 to 90%. The issue in leukemia is quite different. Out of those 90% of patients or so that will achieve remission, almost half, potentially over half, depending on the study you look at, will relapse within the first year. And when you look at the drivers of poor response or relapse, we're learning that they really fall into two broad categories. Certainly, there's others that are being looked at, but the primary ones are, one, the quality of the product. This is a very different drug in the sense that it's a living cell that needs to be infused, it needs to expand, and we're learning that that cell actually needs to persist for some period of time in order to sustain a durable remission. The other major pattern of relapse, which is really, it was quite frustrating when we began to see this, was that, one in which the CAR T cells do all that they're supposed to do, but ultimately the malignancy becomes resistant to the CARs, and that's typically associated with loss of CD19, the protein that is being targeted by the CAR T cell. And so in my work at the NCI, serendipitously, quite frankly, we began to, think about, generating CARs that targeted different surface antigens. And the one that we went after is called CD22. It's another B cell associated antigen. There's a number of antibody drug conjugates that have been developed for CD22. And in 2015, we opened a trial. This was around the time that the earliest experience with CD19 CAR T cells in leukemia and lymphoma was starting to emerge. And we opened a trial with a CAR T cell targeting CD22. So, this is the data from that trial. This trial was opened, as I said, in 2015 as a phase 1 clinical trial, and it turned out to be quite effective. This is what's called a waterfall plot. Each of these bars is an individual patient, and all of those bars that reach the bottom are complete remissions in these patients, in about 75% or so of patients that received the CD22 CAR. This was initially an ALL trial, and initially we enrolled all comers, including patients that were naive to CAR T cells. But we ultimately began recognizing the importance of this therapy in patients that had received a CD19 CAR and had relapsed with CD19 negative leukemia. And there was no difference in response rate. We've certainly learned, we learned a number of other things from this trial. The fact that the details of how these cells are manufactured can make a difference, both in terms of efficacy and toxicity. that this CAR had a similar but slightly different toxicity profile. There was a low incidence of neurotoxicity and a higher incidence of an inflammatory toxicity called hemophagocytic lymphohistiocytosis or macrophage activation syndrome. This is also seen in other diseases like rheumatoid arthritis. It's actually, as you know, highlighting something Clay said earlier, it is a toxicity that seems to not respond to the standard IL-6 anti-IL-6 therapy that's used in the context of cytokine release syndrome. More recently, and this is you know, something that you know, has been quite important, has been you know, taking the CD22 CAR from the leukemia experience and really testing it in lymphoma, and this is really happening currently at Stanford. And what the Stanford team has done is, as we did in the leukemia cohort, was to begin to use the CD22 CAR in patients who receive a CD19 CAR and for whom the CAR treatment fails. And this was published this year. This were the first 3 patients treated. All three of them had failed prior CD19 CAR therapy. One of them actually had received a CAR targeting 2 different antigens, CD19 and CD20. And all 3 of these patients achieved a complete remission. This group will be presenting the updated experience with the CD22 CAR in these CD19 CAR refractory patients. You know, the updated experience looks very comparable to this experience. Some of these patients, you know, at least over 6 to 12 months, seem to be enjoying sustained remission. So, you know, evidence that this CD22 CAR is not only very effective in leukemia, but also highly effective in the setting of chemotherapy refractory and CAR refractory diffuse large B-cell lymphoma patients. So one of the concepts that we began considering early on in our leukemia experience with the CD22 CAR was, although we were able to use it as a salvage after CD19 CAR treatment, you know, we noted that what ultimately happened with a high percentage of those patients that achieve remission with the CD22 CAR was they then lost or dropped expression level of CD22. So we essentially, you know, in a sequential way, took a leukemia from expressing CD19 and CD22 to not expressing either of the 2 antigens. You know, obviously very frustrating. And so early on, we began to consider, you know, how we could potentially utilize a highly effective CD19 CAR and a highly effective CD22 CAR up front to try to increase remission induction rate potentially in lymphoma and to potentially sustain remissions in a higher percentage of patients with leukemia. And so the way that we approached this was to generate these lentiviral vectors, these HIV-based vectors that Clay was talking about earlier, such that we can drive in the same T cell expression of both chimeric antigen receptors. What's shown on the right-hand side there is our ability to do that with a couple of different CAR formats with different co-stimulatory domains. The one that ultimately emerged as the preferred construct is on the left-hand side. It's a CD22 CAR with a 4-1BB co-stimulatory domain and a CD19 CAR that's a version of one of the FDA-approved CD19 CARs. This is mouse data. This is a classic, you know, model that we've utilized in much of the CAR T cell development work. This is a leukemia that we can gene modify so that it glows as it progresses in the mice, and so the color represents leukemia. And in this experiment, what was done was that we took leukemia that expressed either CD19 alone, CD22 alone, or both antigens, CD19 and CD22, the parental cell line. And as you would expect, when you infuse a chimeric antigen receptor that targets only one of those antigens, you end up with progression of leukemia that does not express the targeted antigen, sort of a form of the or a model for the antigen loss that we've seen in patients. On the left-hand side are 3 different versions of the CD19 by CD22 combination CAR T cell. The one on the far left is the one that ultimately emerged as our preferred construct, as I said. And what you see here is the ability for this chimeric antigen receptor combination in a single T cell to eradicate all three subpopulations of the leukemia, those that express both antigens and those that only express single antigens. So we're very excited that, we've now reached a point where we're ready to try this in patients. And so there's a clinical trial that just opened up at the University of Colorado with a chimeric antigen receptor delivery system that will generate T cells expressing both of these. This will be manufactured at the Gates Biomanufacturing Facility. And the goal is to enroll the first patient on this clinical trial in the very near future. And just as a final comment here, certainly, the success with CAR T cells in cancer has led to enthusiasm for the B cell depleting potential of CD19 CAR T cells in non-malignant diseases. I think most of you probably are aware of the fact that anti-B cell antibodies have been effective in high-risk rheumatologic autoimmune diseases, including lupus. And this was a case report that was just published in the New England Journal of Medicine. This was an experience in Germany with a 20-year-old female with lupus, active nephritis, with nephrotic syndrome, pericarditis, as well as other cardiac manifestations, had received a number of different B cell depleting types of therapies, rituximab, as well as other immunosuppressive medications, had failed all of those therapies. The disease activity on the right-hand side here is represented by circulating anti-double-stranded DNA antibodies. And what's shown on the far right there is with a single infusion of a CD19 CAR T cell, this young woman had complete eradication of all manifestations of her disease and a decrease in her double-stranded DNA antibody levels. And so certainly has generated enthusiasm for the potential for this therapy beyond malignant diseases. So, just by way of summary here, hopefully, Clay and I have convinced you that immune effector cells have certainly been a revolution, I think, in cancer care and immunotherapy. And can lead to long-term responses in these otherwise relapse refractory hematologic diseases. As Clay mentioned, there's serious toxicities, ICANS and cytokine release syndrome. We're learning how to manage those, but to do so effectively really requires a tertiary care setting with all of the expertise that Clay highlighted, to do this successfully with the groups at the University of Colorado that have implemented these therapies. There's about 100 immune effector cell therapy centers in the United States. I think our programs are leaders in the region for sure, and the only one with the the only one with the ability to manufacture our own products in the context of these innovative clinical trials. Multiple advances are certainly, under development and testing, with these therapies. Certainly enthusiastic about the potential in solid tumors, although I certainly think there's going to be a number of challenges there. And as Clay mentioned, in spite of the success, both cost and really access worldwide, I think is a real challenge that we have to, take a hard look at. And as I said, certainly interested in potential applications for this therapy beyond cancer. And so as a final slide, I think Clay, really highlighted, the team effort at CU Anschutz and all of the different contributors to our ability to do this at CU Anschutz. Just as a final set of acknowledgements, the NCI is where the CD22 CAR trial was implemented and still continues. And then, much of the current, CAR T cell research work, is being conducted by the folks in my lab and Eric Kohler's lab at CU Anschutz, as well as a number of other labs that, we're really excited to collaborate with around campus. And so with that, I'll stop and turn it over to questions.
That was great. Dr. Fry, Dr. Smith, thank you very much for this presentation today. And I will say, judging by the number of people in the audience, which is higher than usual, there's a lot of excitement around the topic. I'll give our first question to our chair of medicine, Dr. Chopra. He asks, and I think it's related to the fact that, some ICU doctors and specialized hospitalists may see these side effects more frequently than others, but as they become more common, more of us are going to need to know more about them. So can you tell us more about the potential adverse effects from CAR cells and how the patients present, and more probably as importantly, when they present after their therapy?
Yeah, I'm going to turn it over to Clay in a minute. What I will tell you is one of the things that we're learning is that these patients present typically with high fever. I mean, that's typically the first manifestation of cytokine release syndrome. And, I've been humbled by how quickly it can, quite frankly, devolve from high fever to, cardiac collapse, et cetera. And so one of the key components of any program is the ability to take patients that have been treated, and, these patients are outpatient until they develop the fever. They need to come in and they need to be seen immediately and get started on therapy, and that is a key component of the ability to do this safely. Clay, anything to add there?
Yeah, I would say, again, this at the moment requires a lot of experience. We've definitely learned along the way hopefully how to manage this more effectively. But frequently in our patients, it can present as fever when people are neutropenic. So you have to make sure you start antibiotics, because sometimes they actually do have an infection rather than CRS. And it can progress through hypotension, hypoxia, capillary leak, hypotension that does not respond to pressors. And the worst of these is what Terry described earlier, that HLH, where you can add one treatment after another and the patient may still not respond. There's a lot of work going on across the country and the world about how to properly grade when to start anti-IL-6 therapy or steroids. Still fairly controversial. We have our own fairly strict internal guidelines, and then we're trying to collect data on our populations and then analyze that data and see where do we try to draw the line between starting and not starting some of these diseases. The ICANS is also very peculiar. I mean, you can see people that are completely awake talking to you and they cannot come up with certain words, and that's all that you see, all the way up to people having seizures or awake but not responsive. It's a very odd clinical syndrome, and that also, of course, can mimic many other neurologic side effects that we see. And so managing that and diagnosing it and grading it is also driven by a lot of guidelines, a lot of data collection and review in our center.
Comment on that, on the neurotoxicity. It is a really peculiar toxicity. And I will tell you that in pediatrics, although we certainly, in the extreme forms, see the same manifestations, that is seizures and obtundation. The early manifestations in pediatrics, interestingly, can be different. I mean, the expressive aphasia, the word finding difficulties Clay mentioned, we don't see that as often in the younger kids. What we see in the younger kids is actually hallucinations. So it's a really fascinating toxicity. And as Clay mentioned, we don't really understand the pathophysiology of that very well.
Yeah, lots to learn there, obviously. Terry, you mentioned toward the end of your talk there about the use of CAR T cells for other things, and this was other CD19 targets. Can CAR T cells or other sort of CAR fill in the blank cells be used for other non-malignant diseases where there's a target molecule and target cell? And is that being worked on?
Yeah, it definitely is. I mean, there's a lot of enthusiasm. I mean, what I really described was, using the same CAR, to deplete B cells, right? Normal B cells. By the way, one of the expected, toxicities of CAR T cells is that these patients are, essentially B-cell aplastic for sometimes a year or more. In our pediatric patients, we typically replace, antibodies to try to prevent infections. And so, what I described was really, the use of a CD19 targeted CAR to induce, a response in a autoimmune, a patient with autoimmune disease. There are other, genetic modifications that can be incorporated into a CAR T cell manufacturing process, either to target other antigens or to convert a T cell into what's called a regulatory T cell, a T cell that actually can shut down immunity. And there's actually a number of companies and a number of academic programs that are beginning to explore that approach in autoimmune diseases. So for sure, there's a lot of interest in, I would say, increasing the sophistication of the genes that we're putting into the T cells.
That's great. I know we're right at time, but we have one question from our residents I want to make sure we get to because it's very important to them, which is, you mentioned that we're creating the cells here on campus. And Dr. Smith, you mentioned how that allows us to get those cells out to people who couldn't afford them otherwise. How are you using that lab to improve access to care? And what's it going to take to sort of reduce cost overall so we can get these therapies to more and more people?
Yeah, that's a great question. And I think it's something that is really troubles and challenges Terry and I and others in this field, is that it is a very expensive form of therapy. And I know I speak for Terry, both of us, we all trained at places where, we're really committed to seeing an egalitarian and accessible form of medicine. And I think that's one of the big issues. Some of this, frankly, is markup by pharmacy companies that doesn't reflect the actual cost of manufacturing, but other elements. I think at last count, I think there's something like 600 different companies in the cell therapy field right now. I am hopeful and that many of them are trying to find more cost-effective approaches. I think if we can move to that off-the-shelf approach where we're not collecting cells, modifying it on a patient-by-patient standpoint, that is going to be huge. And then don't forget, this is only one part of the broader field that Terry started this talk with of immunotherapy. There are antibodies, there are other forms of therapies that ultimately may end up being as effective and a lot cheaper and more accessible to people. So a really important question, and I think one that we hope our field tackles. And I know, again, I speak for Terry, that we are actively contributing to that as we can.
That's great. Thank you very much. Well, it is 1 after 1. I want to be respectful of your time. So Dr. Fry, Dr. Smith, we have a number of other questions. I will email them to you so we can get some answers for the crowd. But thank you again for the time and for the groundbreaking work that you're doing. I mean, you really are at the forefront of this and keeping our institution at the forefront. So thank you for all the work and thank you for the talk today.
Thank you.
Thank you.